Look closely at almost any ecosystem, and you will see organisms pouring immense amounts of energy into finding, courting, and mating with a partner. Evolutionary biologist John Maynard Smith described this biological imperative as an embarrassment for the theory of evolution. This standard reproductive strategy is called anisogamous sex. It relies on the biological fusion of two differently sized gametes, typically an egg and a sperm, contributed by a female and a male. But nature offers a biological alternative, apomictic parthenogenesis. In this system, females bypass males entirely, cloning themselves to produce genetically identical daughters. This raises a question of efficiency. Why does nature favor a complicated, resource-heavy two-partner system when a streamlined single-parent cloning process is readily available? To see the severity of the problem, we have to look at the generational math. This visualization compares two distinct starting populations. On the left, sexual reproduction requires an equal mix of males and females. On the right, an asexual, parthenogenic population consists entirely of females. Because the asexual group doesn't produce males, which cannot bear offspring, every single individual in that population reproduces. In a single generation, the clonal population produces double the reproductive offspring of the sexual one. As the cycles continue, the discrepancy compounds. The asexual population rapidly If a functional asexual mutant emerges within a population, its numerical advantage should allow it to outcompete its sexual ancestors in a few dozen generations. Yet sexual organisms dominate the biosphere. This implies that specific selective pressures must be actively defending sexual reproduction from being overwhelmed by asexual variants. Evolutionary biologists often look to ecological selection to explain this survival. This is a fitness landscape, mapping how genetic variation aligns with survival. The high peaks represent optimal environmental adaptation, while the valleys represent extinction. Because asexual clones are genetically identical, they are highly specialized. They group tightly on a single peak, adapted to specific conditions but entirely uniform. Sexual reproduction constantly mixes the genome. This recombination scatters a diverse array of offspring across multiple different peaks, ensuring a wider distribution of physical traits. Under the Red Queen hypothesis, environments and pathogens constantly shift. When conditions change, the specialized peak supporting the clones collapses. The sexual population survives because its genetic diversity ensures some individuals are already positioned on newly formed peaks. Recombination creates an essential genetic buffer against these environmental shifts. However, ecological resilience is a long-term benefit. It does not address the immediate problem of a highly fit asexual clone outperforming its neighbors during the time it takes for an environment to shift. Another major theory shifts the focus from ecology to genomic parasites. In this model, self-interested genetic parasites, like viruses or transposable elements, depend on horizontal gene transfer to infect new hosts. These elements can spread more effectively through populations that regularly mix their genetic material. This network visualizes a population's genome. The constant mixing of genes through sexual recombination creates a highly connected infrastructure for these parasites. Parasites move easily through the sexual nodes, but cloning creates a closed loop. The moment an asexual lineage breaks off, it isolates its bloodline, blocking the parasite from entering. While parasites and environmental shifts create powerful selective pressures over hundreds of generations, the twofold cost of meiosis is felt in every single generation. A clone population doubles its frequency immediately. For a parasite or a collapsing peak to counter that advantage, the selective pressure must be equally rapid. Identifying a mechanism that functions the moment an asexual mutant first interacts with a sexual population requires looking at the direct physical interactions between the two modes. This brings us to reproductive wastage, the theory that males themselves act as a biological deterrent against invading clones. When early parthenogenic variants emerge, they occupy the same environment as their sexual ancestors. These new clones will share space, resources, and mating encounters with the parent population. These physical interactions can trigger a specific biological event. A sexually reproducing male may fertilize the asexual female's egg, but because she is a clone, her egg is already unreduced. It contains a full diploid set of chromosomes. This creates a mathematical error. A haploid sperm fuses with a diploid egg carrying 2 chromosome sets, creating a triploid offspring. This extra set causes a catastrophic cellular imbalance. In most species, this triploidy is completely lethal, and the offspring dies. This is the triploidy defense. It suggests that males act as an automatic culling mechanism, destroying the potential offspring of asexual mutants through the act of fertilization. To determine if this mechanism can actually overcome the 50% numerical advantage of clones, we can look to the population genetics models of Mikkel Kreber and Michael R. Rose. In 1986, Kreber and Rose attempted to mathematically parameterize this conflict to see if males truly stop asexual invasion. Their core selection scheme is summarized in Table 1. It defines frequencies and viabilities of genotypes where a sexual allele competes with a parthenogenic allele. The model tracks different egg types, specifically isolating unreduced fertilized eggs, those resulting in lethal triploidy. The researchers focused on variables like male virility, labeled C, and the probability of males successfully penetrating asexual eggs, labeled A and B. They used these parameters to build differential recursion systems, which calculate how allele frequencies change from one generation to the next. Table 2 shows the boundary stability conditions. These equations define the exact mathematical thresholds where the sexual population remains stable and the asexual allele is eliminated. The results showed that for sex to be stable, the combined sexual and parthenogenetic output of the mutants must stay below the fitness of the sexual form. Specifically, if triploidy is the only pressure, sex is only stable if males destroy at least half of all parthenogenetic eggs. But when the researchers ran the final numbers, they found a result that changes how we view the survival of clones. The models show that even when lethal triploidy destroys half of all asexual eggs, the parthenogenic allele is not necessarily eliminated from the population. This phase space graph plots the population trajectories. As the clonal population encounters the triploidy defense, its growth curve flattens. Instead of hitting 0, the trajectory line curves. The asexual lineage survives by moving away from the sexual boundary, stabilizing in a separate area. This happens because male interference acts as an intense selective filter. Only the clones that can avoid male fertilization survive to reproduce. This selective pressure forces the clones to develop rapid reproductive isolation. Any mutation that causes genital tract incompatibility or egg surface sperm exclusion becomes highly favored. Over time, the encounters between the two forms stop resulting in fertilization. The asexual lineage diverges, becoming a distinct, separate biological path. The models of Kreber and Rose suggest that the very weapon males use to destroy clones provides the exact selective pressure needed to drive them into reproductive isolation. Rather than eliminating the invasion, the triploidy defense forces the clones to evolve into an unstoppable new species.